One of the functions of the endoplasmic reticulum is the vesicular transport of the secretory proteins to the Golgi apparatus, where they undergo post-translational modifications so they can be translocated to their final destination. The coat protein complex, or COP2, is a group of cytoplasmic proteins that are essential to the formation of vesicles at ER exit sites. Sec12 is an ER integral membrane protein, which catalyzes the exchange of GDP for GTP in SAR1. The activated SAR1 then binds to the surface of the ER membrane, where it recruits the dimer, Sec2324. This subcomplex now roams the ER until it interacts with and secures the protein to be transported, the cargo protein. Other dimers, Sec1331, are recruited to form a support structure with Sec2324 and SAR1. The ER membrane starts to bend, allowing the support structure to extend into a polyhedron until a vesicle carrying the cargo protein molecules buds out of the membrane. Soon after, the coat proteins detach from the vesicle due to the hydrolysis of GTP in SAR1. The vesicle can now travel from the ER to the cis face of the Golgi, where it will fuse with the membrane and release the cargo proteins into the Golgi lumen. Lipid composition plays an important role in membrane bending rigidity, with conical lipids enhancing membrane curvature. Common membrane lipids include phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, and phosphatidylinositol. When these are cleaved by phospholipases, they produce lysophospholipids, which are conical. So the authors wanted to investigate the impact of lysophospholipids on the formation of COP2 vesicles. To do this, they use the Sec12-4 yeast strain that is known to have disorganized ER exit sites, exhibit low efficiency of activated SAR1 recruitment, and is temperature sensitive. They overexpress 3 phospholipases in the mutant cells to observe the effect of lysophospholipids. They found that overexpressing phospholipase B1, or PLB1, actually helped the Sec12-4 mutant overcome its temperature sensitivity to some extent. Overexpression of PLB2 was lethal at all temperatures. They obtained more satisfactory results with PLB3, which, when overexpressed in the mutant yeast cell, allowed the cells to grow above the temperature limit. However, overexpression of PLB3 in other yeast mutants did not achieve the same results. To get a better understanding of what was happening in the ER membrane, the authors visualized the ER exit sites and conducted mass spectrometry analyses of the lipid composition of the cells and compared them to the lipid composition of the ER. They achieved this by disrupting the cells and separating the ER membrane microsomes from the rest of the cell components. With different reagents and stains, they could visualize the ER exit sites of wild-type and mutant cells. What they found was that the microsomes from the mutant cells had significantly fewer ER exit sites than the wild-type microsomes. However, overexpression of PLB3 in the mutant cells resulted in a rescued response, even at higher temperatures. Mass spectrometry analysis of cell lipids revealed mutant cells with overexpressed PLB3 had significantly higher concentration of phosphatidylcholine than the wild type, and a decreased amount of phosphatidylinositol. Interestingly, when they looked at the lysophospholipid profiles of wild type and 3 mutant cell types, they observed that lysophosphatidylinositol was present in the highest abundance. These results suggested that even mutants with defects in the secretory pathway could be rescued by the presence of increased abundance of lysophospholipids. When the cell lipid profile was compared to the microsome lipid profile, it was evident that some lysophospholipids were more abundant in ER microsomes than in other cell membranes. Another interesting observation was that after budding, there appeared to be a higher abundance of lysophospholipids in the microsomes and even higher still in the vesicles. This confirmed the hypothesis that the COPII vesicles were enriched in lysophospholipids. But why? One hypothesis is that the presence of many conical lysophospholipids reduces the membrane bending rigidity to allow the membrane to form vesicles. This could also mean that the presence of many phospholipids enhanced the binding of COP2 subunits so that budding of vesicles could be induced. To test the binding of COP2 components, the authors generated giant unilamellar vesicles, or GUVs, from a lipid mixture. Then, to observe binding efficiency, they injected fluorescently labeled COP2 proteins into the GUVs with different combinations of lysophospholipids and guanylyl diphosphate. The signal intensity was linearized over the circumference of each GUV, then averaged over time. The resulting intensity profiles showed that the best results were obtained when lysophosphatidylinositol and GMP-PNP were both present, confirming that lysophosphatidylinositol enhanced the binding of COP2 proteins. Next, to test the membrane bending rigidity, the membrane tension of GUVs was measured while being injected with lysophospholipids. An aspiration pipette was also used to generate a tongue, and the membrane tension was measured before and after injection of the lysophospholipids. The results show that the membrane tension decreased and the membrane bending rigidity dropped by an average 26% after injection with lysophospholipids, confirming that increased abundance improved membrane curvature. The results described in the paper all point to the importance of lysophospholipids in making the ER membrane less rigid, enhancing the binding of COP2 proteins, and therefore enabling efficient formation of vesicles. The authors suggest that increasing abundance of lysophospholipids at ER exit sites may be an effect of lipid sorting at curvature locations. Furthermore, the enzymes responsible for remodeling the membrane also contribute to the increase of lysophospholipid abundance by producing more of them.